Hello everyone. Welcome back to Gray's Gyan, where we take one topic from Gray's Anatomy every single day and break it down so simply, even a first-year student can master it. Today's topic is something absolutely fundamental to how your cells function at a microscopic level. We are talking about microtubule-associated proteins, also called MAPs. Now don't let the big names scare you. By the end of this video, you will understand exactly what they are, what they do, and most importantly, you won't forget them in your exams. So let's get started. Before we talk about MAPs, we need to quickly understand what microtubules are. Think of microtubules as the railway tracks inside your cells. They are tiny, hollow tubes made of a protein called tubulin. These tracks run throughout the cell and serve three main purposes. 1. They give the cell its shape. 2. They act as roads for transporting materials inside the cell. 3. They are essential during cell division, helping to pull chromosomes apart. Now here is one important thing to know about microtubules. They have two ends. A plus end and a minus end. The plus end is the actively growing end. And microtubules have a property called dynamic instability, meaning they are constantly growing and shrinking. Like a railway track that builds and dismantles itself depending on where the train needs to go. Now, what controls all of this? What proteins bind to these tracks and manage everything? That is exactly where microtubule-associated proteins, our MAPs, come in. Microtubule-associated proteins, or MAPs, are proteins that bind to assembled tubulin. That means they attach to microtubules that are already formed. Gray's Anatomy tells us that these MAPs are concerned with two broad things. Either structural properties, or motility, which means movement. So we can divide all MAPs into two big families. Family number 1, structural MAPs. Think of them as the builders and organizers. They form cross bridges, connections, between adjacent microtubules, or between microtubules and other structures like intermediate filaments, mitochondria, or the plasma membrane. Family number 2, transport MAPs. These are the motor proteins. They are found wherever movement happens along microtubule surfaces. Like cargo transport, cilia bending, flagella movement, and spindle movements during cell division. So remember, structural MAPs equals organization and stability. Transport MAPs equals movement and motility. Simple as that. Now let's go a little deeper into structural MAPs, starting with a very important subgroup, the plus-end associated proteins. These are proteins that specifically associate with the plus end of microtubules. Remember, that's the growing, active end. What do they do? Two main things. 1. They regulate dynamic instability, meaning they control whether the microtubule grows further or starts to shrink. They are like traffic controllers for the railway track. 2. They manage interactions with other cellular substructures. They guide the growing tip of the microtubule towards specific targets inside the cell. Think of plus-end MAPs as gardeners tending to the growing tip of a plant, deciding which direction it grows and when to stop. Now this section is extremely high yield for your exams. So I need you to really pay attention here. MAPs found specifically in neurons include several important ones. Let's go through them one by one. MAPs 1A and 1B. These are present in both neuronal dendrites and axons. So MAP 1 is everywhere in the neuron. Memory trick, think of 1 as the number 1, the all-rounder. It's everywhere. MAPs 2A and 2B. These are found chiefly in dendrites. Only dendrites. Memory trick. MAP 2 has a curly top, just like a branching dendrite. Tau. Tau is found only in axons. Only axons. Memory trick. TU stands for transport axon unidirectional. And MAP 4. MAP 4 is the major microtubule-associated protein in most other cell types, meaning non-neuronal cells. So MAP 4 is the common person's MAP. Let me repeat that quickly. MAP 1, dendrites and axons. MAP 2, dendrites only. TU, axons only. MAP 4, non-neuronal cells. And one very important clinical point, TU is directly linked to Alzheimer's disease. When TU protein becomes abnormally phosphorylated, it forms neurofibrillary tangles, which are a hallmark of Alzheimer's. So knowing TU is not just exam knowledge, it's real clinical medicine. Now let's talk about why structural maps matter, their significance for the cell. Gray's anatomy tells us that structural microtubule-associated proteins are implicated in 3 critical processes. Microtubule formation. They help build new microtubules by stabilizing tubulin as it polymerizes. Microtubule maintenance. They preserve existing microtubules and prevent random breakdown. Microtubule disassembly. They also trigger breakdown when the cell needs to reorganize, for example during cell division. Because of these 3 roles, structural maps are critically important for cell morphogenesis, meaning how a cell develops its final shape. Mitotic division, the process of cell division. Regulation of cell shape, how the cell maintains and changes its form. So basically, without structural maps, your cells would lose their shape, couldn't divide properly, and the entire cytoskeleton would collapse. They are the invisible architects of the cell. Now let's talk about transport maps, the motor proteins. These are the stars of the show when it comes to movement inside the cell. Gray's anatomy says, transport maps are found in situations in which movement occurs over the surfaces of microtubules. Examples include cargo transport, cilia and flagella bending, and mitotic spindle movements. The 2 most important motor proteins are kinesin. Kinesin moves towards the plus end of microtubules. In neurons, this means it carries cargo from the cell body towards the axon terminal. This is called anterograde transport. Think of kinesin as the delivery truck going away from headquarters. Dynein. Dynein moves in the opposite direction, towards the minus end. So it brings cargo back to the cell body. This is called retrograde transport. Dynein is also responsible for the beating of cilia and flagella. Think of dynein as the truck bringing things back to headquarters. Memory trick. Kinesin goes kinetically outward. Dynein drags things back in. Both kinesin and dynein are powered by ADP hydrolysis. They are literally molecular machines walking along microtubule tracks, carrying cargo. 2 more transport-associated proteins we need to know about. First, dynamin. Dynamin is involved in endocytosis. What does that mean? Endocytosis is the process by which the cell swallows molecules from the outside. Here's how dynamin works. When the cell membrane forms a pocket around something it wants to bring inside, a vesicle forms. Dynamin wraps around the narrow neck of this vesicle and pinches it OFF, like tying the knot of a balloon. This releases the vesicle into the cell. Think of dynamin as the knotter of endocytosis. Without it, the vesicle can't detach. Second, kinetochore proteins. These proteins assemble at the chromosomal centromere during mitosis and meiosis. Their job is to attach chromosomes to spindle microtubules, basically acting like seat belts that strap chromosomes onto the spindle. And very importantly, some kinetochore proteins are responsible for chromosomal movements during anaphase, both mitotic and meiotic anaphase. So they don't just attach, they also drive the movement of chromosomes to opposite poles of the dividing cell. Alright, let's do a lightning-fast recap before we close. Maps are proteins that bind to assembled microtubules. They either help with structure or movement. Structural maps form cross bridges. They include plus-end proteins that control dynamic instability, and proteins like MAP1, MAP2, TU, and MAP4 in different cell locations. Transport maps are motors. Kinesin goes toward the plus end, anterograde. Dynein goes toward the minus end, retrograde. Dynamin handles endocytosis. Kinetochore proteins attach and move chromosomes during cell division. And functionally, structural maps are critical for cell morphogenesis, mitotic division, and regulation of cell shape. That's the complete picture of microtubule-associated proteins from Gray's Anatomy. If you found this video helpful, please like it. It really helps this channel reach more medical students like you. Subscribe to Gray's Gyan if you haven't already.